All right, it is the top of the hour, so we'll go ahead and get started. I'd like to welcome everyone to the American Floral Endowment webinar series for Thrips and Botrytis Research. We are pleased to announce 2 new sessions in the series this fall, which will be presented first in English. Our Spanish sessions will follow in the coming weeks, and those dates are to be announced. Today's session is on screening biological control agents for effective Botrytis blight management. I'm your webinar host, Dr. Garrett Owen, Assistant Professor of Greenhouse and Controlled Environments at the University of Kentucky. Today, we'll be hearing from Dr. Jim Faust, Associate Professor of Floriculture Physiology at Clemson University. Today's session will be presented live in English. Jim Faust will join us following the presentation for a Q&A session. We ask that you submit any questions through the Q&A feature at the bottom right of your screen. We will answer as many questions as possible before the end of the hour. These live sessions are exclusively for the supporters of the Thrips and Botrytis Research Fund. The series is being recorded and we will release this recording to the public in 2022. These presentations and materials are copyrighted and should not be distributed or published without AFE's written permission. AFE is the national nonprofit organization that funds scientific research and identify and solve challenges within the floriculture industry. In 2021, AFE is celebrating 60 years of providing for the future of floral industry. In 2017, after listening to important industry feedback, AFE established a special research fund to aggressively address the control and management of thrips and botrytis. This campaign has produced several webinars and vital information for our supporters. Past webinars can be found on our YouTube channel, and more information can be found at endowment.org. Again, that is endowment.org. The American Floral Endowment and all researchers would like to thank all organizations who have contributions in support of this important initiative. Today's speaker is Jim Faust. Jim Faust, like I had mentioned, is a professor in the Plant and Environmental Sciences Department at Clemson University, where he teaches floriculture and hydroponic courses and conducts a research program focused on floriculture greenhouse production. Current research projects focus on botrytis management strategies, chrysanthemum leaf necrosis, and international cut flower production physiology. Jim, along with John Dole, have recently edited a new book entitled Cut Flower and Foliage, printed by CAB International Publishing. Jim, welcome, and thank you for joining us today.
All right, Garrett, thanks for that introduction. It's a pleasure to be here today to give you a bit of a sneak peek into the research that we're doing on disease management alternatives for botrytis blight. We're calling it a sneak peek because we're 1 year into a 3-year project, and we have some interesting results that I think that you'll be interested in seeing, and we'll continue to communicate our results as over the next couple of years as they develop. So what we've been doing is looking at various products, most of them being biological control products that we would consider alternatives to traditional chemical fungicide treatment for botrytis. And so the process that we're going through this whole project is to look at basically 3 species, petunia, rose, and gerbera. And then initially we started screening 15 products, and we're kind of adding those as new ones come to light, and we need to consider those. And then the idea is once we've screened those 15 for each of these species, we then take the kind of the winners, the ones that are looking the most promising for commercial production, and then focusing on those products. So what we'll show you today primarily is this work that we've kind of completed the initial screening on petunia, and then we've also, because we've seen some pretty interesting stuff on one of the products in particular, we have started to do some focused work on one particular product on roses. The reason we use these three species is, of course, they're important, but they also give us really three different types of flowers. What we are finding is that petunia, we tend to see the best results and they tend to be easiest to control. Botrytis, simply because you have a single petal that you can actually get whatever product you're applying, you can get it on those petals fairly easily and get good coverage. Rose is a little more complicated because you have an array of many petals. During the production cycle, you basically are just spraying the back petal of the bud. So just those outer petals are being treated. And then as the flower opens, the inner petals are exposed. Of course, spores can land on those inner petals, and you don't, and those are harder to actually get an active ingredient on. So it's a little bit harder to manage and apply these products effectively on rose than petunia. Gerbera, where we've had the biggest challenge in our experiment so far, is I think because of, again, the habit of the plant. With rose and petunia, you have the flowers being formed at the top of the canopy where we can spray pretty effectively. With Gerbera, the flowers forming down in the underneath the canopy, where there's often, I think, a lot of Botrytis spores floating around. Those buds can be inoculated with Botrytis at an early stage. Then the flowers rise up above the canopy. You actually start to get to spray them fairly effectively. But if the infection has already occurred, then the spray applications are not going to be as effective, because many products affect the germination of spores, not so much of the control of the mycelial growth in the tissue. So Gerbera has been by far of these three species the most difficult to control. So if we look at the 15 products that we're currently evaluating, you'll see they kind of fall into 7 different groups. We have 4 bacteria, where the active ingredient is actually a bacterium. We have 4 fungi. Again, the active ingredient is a fungus. 2 bioproducts, meaning these are things that are produced by plants or bacteria. We have 2 nutrients that we're looking at. You're probably familiar with our work on calcium, and so we have added a different form of calcium that is considered organic, where it is a calcium ion that's been attached to a protein molecule. And so it's basically a different way to apply, a different form of calcium that can be applied. And then we've added silicon, since there has been some documented evidence of silicon working on some fungal pathogens like powdery mildew. We have 1 plant extract, 1 synthetic biostimulant, and then 1 systemic acquired resistance inducer. And so these are the first ones we're trying. And then these are the product names. These are the active ingredients in those products. So there are different ways that you can evaluate botrytis, and we've done 3 different ways, actually. The way that we have kind of focused in on that we think is the most useful for initial screening is the method that we're using here today. And that is, we grow plants in a greenhouse as clean as possible. These are petunias. We harvest the flower. We spray the face of the flower that is open, allow it to the spray to dry over 24 hours. The base of the flower is put in water so it doesn't wilt. And then we come in and inoculate the face of the flower, the open face, with Botrytis spores. And then we place that flower in containers that we can seal the lid on and create a 100% humidity environment. So we call these humid chambers, which allows the Botrytis spores to germinate and grow very quickly. So then over the next 3 days, we do visual evaluations of the flower at basically 1, 2, and 3 days after inoculation. And that's the data that we collect. The other ways that we have done and can do, but don't think it's as useful, we have done Petri dish studies, where you have the active ingredient and Botrytis spore and no plant. So we think that's actually less useful. The other way that is interesting and but is a little bit more complicated is to spray the plants in the greenhouse, kind of like a grower would do, where you grow the plants and spray them every week with these compounds. And in those situations, again, it's a little bit more complex interpreting the data. So we've decided just to go with these single flower treatments followed by inoculation. So this is our simple Botrytis blight disease severity scale. So as the flowers, as the Botrytis grows on the flowers, we have a ranking that we can quantify flowering, or excuse me, quantify disease infection, not flowering. And this, just to give you an example of the data that we're collecting and what these plants look like, we actually put 6 flowers in each of the tubs and they're a combination of 6 different treatments. So this is a look inside one of those tubs 24 hours after inoculation. You see potassium silicate, a product called Pure Crop, the On Guard calcium, a product called Zivion, and a Trichoderma species that we have that is experimental because we have isolated it and are using it for inoculation, and then the control. So you can already start to see after 24 hours some of these treatments starting to look a little bit poor, but 48 hours later you can see these 4 are not looking so good. And then after 72 hours, basically the flower petal has completely disintegrated and is just lying flat up against the styrofoam base. And then you can see in this same container, even though these 4 really look terrible, you have 2 treatments that look really quite good. This would be the calcium and this would be Zivion. And so pretty quickly, you open up a bunch of our tubs with these treatments and you can pick out the treatments that are looking really promising and that we start to get pretty excited about. So we have a single slide showing you the results of our petunia studies, and this is a lot of work going into one slide, but it sums it up nicely. We have our Botrytis blight severity scale, so 0 meaning no infection, 9 being a completely dead flower. And then we have our treatments. These are 10, we have the control here, and 10 treatments that show no effect over 3 experiments. So again, this has been repeated 3 times, and none of the 3 times did we see any value in or any effect of these products. We saw on 2 products occasional benefits, meaning of the 3 studies, we did not have a significantly positive effect all 3 times. And so this kind of the sum of the 3 experiments, yes, it is significantly different, hence the B's versus the A's here, but it wasn't completely consistent. Then we looked at the best performing products, and the first to point out, we have 2 fungicide control groups. So we have Captan and we have Maravis Prime, which is Pitiflumetafim, which is relatively new to floriculture. And so there's not a lot of resistance out there yet. So you can see that was particularly good. We have an uninoculated or non-inoculated control, which has essentially no disease. And then we have 3 products here that we applied that were as good as or comparable to our fungicide treatments. And that would be Howler, which is a bacterium, Zivion, which we'll go into a lot of detail momentarily about this product, and then again, the calcium treatment that you've seen plenty of data from us already showing the benefits of calcium. This kind of reiterates that calcium works in a different form than what we've used previously. And it shows that these other products are, kind of in that same ballpark of efficacy as calcium and in the same ballpark of efficacy compared to the chemical fungicide treatments. So going forward, then our top performers were these three. We've already done a lot of work on calcium, so we are not looking at this in more detail. Howler has really stood out here in the recent month, and so we will do some more detailed work with this. Zivion, we started seeing some real benefits of several months ago, so we've actually made some progress beyond the initial petunia studies, and from now on I'll be referring to it as natamycin, the active ingredient name. So really the rest of the talk here, I'm going to go into some detail on this product because it's new to agriculture, and it's, to my knowledge, has never been used on floriculture crops, and shows some real promise. So we acquired this product through a product called Zivion, which is labeled for mushrooms. So it suppresses a disease called dry bubble disease, or it's a verticillium. And so that's how the form that we used. Where you would actually see natamycin more commonly is if you purchase cheese products and you look on the back of the label. This is a very common food preservative used in the food industry. So if you look closely at the label here, besides all the cheese active ingredients, you see natamycin, a natural mold inhibitor, listed in here. And so this has been used for decades in the food industry. It's considered quite safe, and it, again, it's a preservative, and so I'm going to give you some list of facts about it, so you know a little bit more about it before you perhaps consider looking to try some in some internal evaluations. So it is considered a natural biofungicide. It is also named pimaracin, and you'll see this sometimes in the online and in the literature. So it was originally found in South Africa in the 50s. It is a fermentation product of Streptomyces natalensis, which is a soil bacteria. The way it works is by inactivating the germinating fungal spores by blocking cell wall formation. So as the germ tube starts to form, the cell wall on that doesn't form well, and so then the fungus dies. So once you get past spore germination, it's not particularly effective. It does not affect the mycelial growth. So again, it's not a curative type of product, it's a preventative. It does not affect bacterial growth at all. It is a widely available product in the food and the pharmaceutical industry. It is available in North America. It is also available in South America. And it is used in, we've looked, and it is used in food products in South America as well as in North America. It is an antifungal additive to food products. Most commonly, we see it in cheeses, but you also hear of it in yogurt, salami, bread, tortillas, cake, and other things. It is also in the pharmaceutical industry used to treat fungal eye infections. So again, this is something that's quite safe for humans, which is also an exciting thing to consider when we start to apply it to flowers. That we know we're applying a very safe product. So it has broad spectrum efficacy, effect against many different fungal organisms, including botrytis, powdery mildew, pythium, and so on. It has been EPA approved for pre and post-harvest agricultural applications as of 2012. It also has organic status in Canada. In horticulture, it's a biofungicide, approved in California. Where we've seen it most often, the best commercial product, as I showed you, is that we found is Zivion, which is is used for mushroom production. and has been labeled since 2014. It does have a 4-hour reentry interval. It also is labeled for post-harvest use in various fruits, stone fruits, citrus, cherries, avocado, kiwi, mango, and pomegranate. It has been used as a pre-plant dip in strawberry. And what is interesting is these all have something in common. These mushrooms are grown in the dark, post-harvest is in the dark, and as a pre-plant dip, you're doing that before production. The reason is because natamycin is sensitive to UV light, and so it can't be used outside. Also, fungicide resistance has never been reported, and it's not expected. It's not expected that fungi will develop resistance to this product. And as I mentioned, it is sensitive to UV light. And I think that's the reason why even though it's been available since the 60s, it has not been used much in outdoor field agriculture because it would break down quite quickly. I guess you could spray it on a product in the field at sunset and get some efficacy overnight. But where it has real promise and where we really could use some valuable products is in greenhouses. If you have a greenhouse that sufficiently blocks out UV light, which many greenhouse glazing materials do, it could be used. It can be used, pre-transplant to treat young plants. It can be used in post-harvest treatments, where again, sunlight is not in any large intensity going to be shining on the plant. So there's real potential with this, and it really has not yet been tapped in our industry to the best of my knowledge. So there's not a lot of research in the plant literature, but this is one study that was done recently, 2018, looking at natamycin pre-harvest dips on control of anthracnose in strawberries. And what you can see here is, the untreated control, water control, and then these guys right here, these treatments are the natamycin at 500 to 1,000 parts per million. And you can see that control level is similar to other fungicides and tank mixes, and actually better than pitiflumethofen by itself. And so again, it's looking as good as chemical fungicide treatments in this study. So, and again, disease severity on this scale. So a 0 is good and a 4 is bad. So see good treatment effect in this study. So we got excited about this and said, well, let's look at roses quickly. And so we have done, we're in the process of, we have a trial in our lab right now, and we've completed one earlier in the summer. And so what we did was we took natamycin dips, and we do a 15-second dip. That's what we've done with calcium in the past. And we use 4 rates, as you can see here. And we did them with and without capsule. We have a Captan control. We are treating flowers that have already been harvested in South America, gone through the post-harvest chain. We buy them locally from a distributor, and so they're, and then we treat them. So we don't know what's been on them previously, but we treat them once we acquire them at the local wholesale flower distributor. And again, we dip them, and then we let that flower in a vase, allow it to dry out, and then inoculate the top of the flowers with all the open petals being exposed with a spore suspension. I'll show you data on 2 varieties that we've done, Jessica and Tiffany. We have Orange Crush in progress, and we looked at those yesterday. And again, the treatments that have looked good with Jessica and Tiffany are standing out pretty obviously on the Orange Crush also. So this is inside of our humid chamber, where you can see a mix of the 2 varieties. This is day 5 after inoculation. And so you can see there's some really bad looking flowers in here and some nice clean flowers. Some of those are the controls, some of those are treated. And then these are our data for Tiffany after 5 days. Again, our natamycin treatments with and without capsule, control on the left, non-inoculated control on the right. And the treatments that are significantly different than this inoculated control on the left are the 250, 500, 750 natamycin treatments. So it looked good. Capsule is kind of, we're not using capsule in the current study. There was no consistent benefit of having capsule in there, so we taken it out. This is what some of the flowers look like after 5 days. The control on the left, severe infection on a lot of petals. On the right is our natamycin dip, looking much better on Tiffany. On Jessica, the results weren't quite as good, but still we had, 2 of the natamycin treatments really stood out, 500 and 750. And as you can see here, the inoculated control looking poor. The natamycin treatment is not perfect for sure, but it is an improvement. So again, it certainly is working and giving us some resistance to botrytis infection. So that's what I have to show you. That's our sneak peek update. So our conclusions are that most of the biological controls we've tested so far have not been effective, unfortunately. And so we have had 3 products that really, as I showed you, had consistent, every time we use them, you got a significant benefit. And those being, again, calcium that we've seen, howler, which is if I hadn't mentioned, is a Pseudomonas bacterium, and natamycin or Zivion. Okay, and so again, we have studies continually in progress, and so we'll keep you updated as we learn more. And just to go back to at the introduction, Garrett mentioned a book that John Dole and I recently edited and co-wrote. And if you're in the cut flower industry or foliage industry, I think it's useful. So I just wanted to show you that. We're pretty excited about it. We think it's there's not a lot of good international cut flower books, and we think it's the best one out there. So, and it should have a nice impact internationally. So with that, we'll turn it back to Garrett and take some questions and try to provide some answers.
All right, thank you, Jim. That's a very interesting presentation and nice sneak peek into your current research and ongoing research. So at this time, if you have any questions, please type those in the chat at the bottom of your screen. It should be labeled Q&A. So we'll give a couple minutes so that if you have any questions, you can get those typed in there. So Jim, as you were going through your presentation, some questions popped into my mind, and those, a couple of those I'd like to go over if we have time. But the first one is, why are most of the biocontrol products not seeming to work?
Yeah, mostly they've been a bit disappointing, and we don't know exactly why. I guess we have a few ideas. One is, when we do our inoculations, we're really overwhelming the plant. We're applying a lot more spores than you would expect to get in a greenhouse. So it could be that the dose is just so, it just overwhelms them, that you would get some effect if the dose were not so high. Although you would like to see an effect with a high dose, but we that might be one reason. The other reason is that especially the living biological products, the concern with them is how well they actually, do they become established on the flowers themselves? And we're somewhat skeptical that how well that happens in many cases, where if you have some confidence that if the spores come in contact with these biologicals, that the biologicals will do something. But but we're not expect some of these biologicals, we're not sure how long they actually live on the flower petals. Once they dry up, we expect they don't live, some of them don't live so long and don't colonize the flowers so well. And so therefore, the period of time that they can actually be effective is fairly limited. So it's not a good answer, but that's the best we have so far.
All right, I appreciate that. So it doesn't seem to be any other questions coming in so far in the chat or in the Q&A. So I'll go ahead and take the opportunity to ask a couple more questions to give everyone some more time. So another one based off of what you had presented, can you tank mix calcium chloride or the On Guard calcium product that you had mentioned with natamycin?
Yeah. So yeah, we expect that we won't have problem tank mixing natamycin with other products. We have our current study that we have going on actually is looking at that specific thing where we've tank mixed calcium and natamycin. So we don't have results yet, but we're hoping that we'll see an additive effect so that we can again use this as a component of a flower dipping treatment. So again, you might have a multiple products in a dip or a spray that give you an additive effect. So you get some benefit, more benefit than if the products were applied individually.
Okay. And with your research, you had mentioned dipping the flowers, but can this actually be sprayed in the greenhouse?
Yes. So on the petunias, we did spray treatments. On the roses, they're dips, and there's and we would expect both would work. Again, because the product is UV sensitive, it might be a better treatment to do in the evening as opposed to in the morning. You probably need to, in your greenhouse, see if you can measure UV light, because it will certainly inactivate it. But again, it could be possible as in a pre-plant sort of treatment. If you're bringing in cuttings or young plants that you expect to have be infected, it's possible to dip cuttings, or again use it in a post-harvest environment. So I think there's possibility pre-harvest, post-harvest, production in the greenhouse, spray, drenches. I think all those things are possible.
Okay, excellent. And if a grower was interested in using natamycin, where would they be able to purchase it? Is it commercially available to be purchased?
Yeah, it's certainly available to the food industry, and it's in the U.S. And again, we've seen it in food products in Colombia in South America, so we expect it's available there also. In the U.S., we know it is labeled for mushrooms as Zivion, so that product is available. We don't know of any products that are currently labeled for flower production, so this is something that we need to start pursuing so that this could be useful for our industry in the future. Yeah.
All right, thank you. So it doesn't appear that any more questions are going to come through the chat or through Q&A. So with that, I think we're going to go ahead and just wrap up today's session. So I'd like to thank everyone for joining us today for another session on Thrips and Botrytis Research. If you have any additional questions or would like to know more information, please contact AFE's research coordinator, Dr. Terrell A. Nell at tnell@afeendowment.org. Again, that is tnell, as in TNELL, at afeendowment.org. For future research updates and other floral industry resources, visit endowment.org. If you have not done so already, please sign up for part 2 of the 2021 Thrips and Botrytis series that will cover Magnificent Marigolds, a plant-mediated system to enhance thrips IPM, on Tuesday, October 12th at 1 p.m. Eastern Standard Time. Again, that's Tuesday, October 12th at 1 p.m. Eastern Standard Time with Margaret Skinner of the University of Vermont. Once again, these presentations and materials are copyrighted and should not be distributed or published without AFE's written permission. The video series will not be released to the public until 2022. Sponsors and supporters will have access to the edited recordings at the end of this series. We ask that you please complete a brief survey about today's session. Your feedback will help us continue to improve our webinars and how we present this research in the future. Thank you again for joining us today, and we hope that you have a great day.